Hello, everyone. Dr. Johansson here. We are going to talk about macromolecules and connect uh, macromolecules with some of the topics we will be studying in cell biology. Hopefully, most of this is review. It's mainly terminology that you need to know. But of course, I will show you the learning outcomes at the end of the talk, but this is the basics of where we're going. So let's just start with the macromolecules of the cell. I'm trying to get this to go away. Okay, so four main types of macromolecules. We will talk a little bit about carbohydrates, nucleic acids, lipids, and proteins. So let's start with carbohydrates, also known as sugars, but they're not all sugars. Okay, so hopefully you remember this general structure of carbohydrates. Carbohydrates stand for basically carbon and water. That's the general molecular structure. So when you're looking for a carbohydrate, you're going to notice lots of OH groups, um, as well as carbons and hydrogens. And most of the time, we represent carbohydrates as this hexamer. There are carbohydrates we'll look at in DNA and RNA that are more of a pentamer. And then when you combine carbohydrates, you can get these massive molecules such as starch. Um, you can get cellulose, which is found in plant cell walls. But what we're most concerned about with carbohydrates is that they're important in modifying proteins and lipids. And so this very busy slide, don't get overwhelmed. There's just a few key points I want you to remember. So first key point is carbs can modify proteins or lipids. And this is done in both the ER and the Golgi. So when we talk about protein synthesis, protein production, we will talk a little bit about the modifications that happen in the ER and the Golgi. Um, Same happens with lipids. So over down here, the bottom of the screen, these are what we call glycolipids. And this whole process is shown up here. It's the word glycosylation. So glycosylation means adding carbohydrates to another molecule, such as a protein or a lipid. Over most of these pictures are carbohydrates added to proteins. So these are membrane-bound proteins, and then all of the nice colored shapes above it are different types of carbohydrates. You do not need to know which types each of these are. You don't have to identify them. Again, the important concept is that carbs can modify proteins and lipids. This is essential for their functions. This happens in the ER and Golgi, and um, carbohydrates and lipids. What was I going to say? I guess important for their function, for some proteins, important for their ability to fold and to be stable. Oh, that's what I was. So not only function of whatever is being glycosylated, excuse me, but it's also important for cell signaling and cell recognition. And we will be touching on some of these topics later in the semester. So, carbohydrates, sugars, glycosylation is that most important word to know, understand. And as we had talked about the different parts of the cell, understand that these modifications, we should write modifications, happen in the ER and Golgi. A lot of carbohydrate modifications start in the ER and are finished in the Golgi. Again, something we'll come back to later. Lipids are the other macromolecule that we're really just touching on this semester, but we will talk about membranes and fluidity of membranes. So the most important macromolecules for lipids that I want you to know is to remind you about phospholipids and cholesterol, because these are important in the cell membrane. Triglycerides are really for energy storage. And the difference is you can see three, what we call fatty acid tails on triglycerides. Phospholipids always have two. So here is an example of a membrane. And we will talk a lot more about membranes, but remember they are bilayers. So some terminology to make sure you know. And we will talk about saturated and unsaturated. So remember that lipids are hydrophobic and they're made up of primarily carbons and hydrogens, very few oxygens. So down here at the bottom are two examples of fatty acid tails. Saturated means all carbon-carbon single bonds. You can see here. And it also means maximum number of hydrogens. So it's saturated with hydrogens. An unsaturated fatty acid is going to have at least one carbon-carbon double bond. And so when you make a double bond, like up here, if we made a double bond, we would lose two hydrogens. Carbon can only make four bonds total, so you would have 1, 2, 3, 4, and the same with this other 3, 4, 2, 1. So the importance of saturated versus unsaturated is in the fluidity, which again we'll talk about soon, but saturated can pack tightly together, making the membrane less fluid, less movable, and you can see that up here. Unsaturated has kinks in the fatty acid tail, which is shown here. And they pack much more loosely. So more space between the fatty acid tails. And this will make a membrane more fluid, things can move around in it more easily. So again, focusing on these different um, vocabulary words. All right, so that's carbohydrates and lipids. The other two macromolecules that we're really going to focus on are nucleic acids and especially proteins. So a term I want you to make sure you understand is the central dogma. Sometimes this is called the central dogma of gene expression. And gene expression for us is going to mean going from DNA to RNA to protein. So first, excuse me, um, part of central dogma is DNA replication. We're going to talk a little bit about this, excuse me, when we talk about cell cycle. But our focus will be more on transcription and translation with respect to the central dogma. So please remember that transcription is DNA to RNA. And it's just a small segment of DNA that is copied. Not everything. Copying all DNA is DNA replication. And we call this small segment of DNA that's copied a gene. So replication and transcription happen in the nucleus. When that RNA is made, remember that RNA is processed, which means it gets a 5' cap, a 3' poly A tail, and the introns are spliced out. So this would be a mature mRNA that's ready to move into the cytosol for translation. So translation is RNA to protein, and this is happening on the ribosomes. Ribosomes can be free-floating in the cytosol or can be found on the rough ER. So central dogma, make sure you know the names of the processes or the steps. DNA to RNA is transcription. RNA to protein is translation. And this is important because we'll talk a lot about proteins. And proteins are the doers of the cell. So we need to understand how to make these proteins. All right. So coming back in general, understanding the differences between DNA and RNA, understanding that DNA is called deoxyribonucleic acid. So that's where D and A comes from. And this is on the sugar of a nucleotide. And I'll show you that a little bit later. Couple things to remember about DNA versus RNA. RNA uses the nucleotide uracil. DNA uses the nucleotide thymine. DNA is normally double-stranded and held together by hydrogen bonds. RNA is generally single-stranded, but I want you to also understand that RNA folds into lots of different structures, and that also involves, excuse me, hydrogen bonds. So big picture there. I want to focus on proteins, and then we're going to come back to a couple really important nucleotides. So proteins are made of amino acids. Amino acids have an amino group, which is an NH. We're going to put 2 here. Sometimes you see it as NH3 plus. The acid part is this carboxyl group, which, oops, sorry, COOH plus another, excuse me, OH there. Oh, that's not right. Sorry about that. C double bond O and OH is the carboxyl. Get this out of here. This is the carboxyl part. I know it's kind of hard to see. And then amino acids have unique R groups or side chains, and I'll show you those in just a second. Amino acids are connected and form peptide bonds. I'm sorry, I have a bit of a cold. Between the amino acids. So proteins are a long string of amino acids connected by peptide bonds. And then protein folding, the concept of shape equals function, we will talk about in a couple chapters. Again, remember these are made during translation. By the ribosomes. And here's just a reminder, amino acids can have different proteins. So, excuse me, they can be hydrophobic. They can have charges. They can just be polar. Remember, these are all hydrophilic properties. Excuse me. And so when we talk about folding, protein folding, that's where the amino acid side chains. So if you look the O, sorry, I'm not saying this very well. This part is the same for all of these amino acids. And these are the R groups that are different and that give the different amino acids different properties. Okay. So again, know these terms, transcription, translation, know where they happen in the cell, what the starting material is, what the product is. Because it's important to understand the concept between genome versus proteome. So genome is all the genetic material in a cell. Proteome is the specific proteins that are expressed. And we have lots of different types of cells. So remember that all your cells have the same genome, same DNA, right? Because you started as an egg and sperm. Once they combined, they created all these different cell types, but they have all the same all the same DNA, but what makes them different is which proteins are expressed. So not everything in your genome is expressed. Fat cells are going to express different proteins than muscle cells because they have different functions, different needs. Um, okay, let's go back to nucleotides. These I want you to see are RNA nucleotides. So yes, these could be incorporated into RNA during transcription. ATP, you probably are familiar with as the energy molecule for the cell. GTP, excuse me, you may be less familiar with, but you will see GTP a lot in cell signaling, not cell like cell, but cell signaling. And it's a way to activate and inactivate proteins. We're not really going to talk about ATP as an energy molecule. Instead, we're going to talk about something called phosphorylation, which means taking a phosphate off of ATP and adding that phosphate to another molecule. And the enzyme that does phosphorylation is called a kinase. So this is an enzyme that phosphorylates other proteins. And we're really going to hone in on that structure equals function. So when a protein is phosphorylated, the structure is changed, and so its function is changed. So phosphorylation can also activate or inactivate proteins, and we'll talk about that. Want you to be sure that you understand that this T stands for tri, and we're talking about the phosphates. So it equals 3 phosphates. And most of the time, I'm going to draw a phosphate as a P with a circle around it. When we make a DP, D stands for di, which means 2. 2 phosphates. And I'll show you in a minute. M means mono, which is 1 phosphate. Processes that are very important for macromolecules. How are macromolecules made? And how macromolecules are broken down. So dehydration synthesis. Synthesis means you're making something. Dehydration means you lose water. So in order to make a bond, for example, a peptide bond, let me go back here, right? So this dashed line, this peptide bond that is connected or helps connect amino acids, that's made by dehydration synthesis. So that means there's an enzyme here that's not being shown that brings these two molecules together, makes a covalent bond, and in that process, water is lost. Water is lost. Yeah. Um, so that's dehydration synthesis. Hydrolysis, on the other hand, means using water to break something. So lysis means break. So in this case, you have another enzyme. We always use enzymes to make these reactions happen. And this enzyme is going to use water to break that bond, right? So now we have two separate molecules, one, two, where here we started with two separate molecules and ended up with one. Now we're breaking things down. We're starting with one molecule, breaking it down into two separate. And that's hydrolysis. So using water to break a covalent bond. Where this comes important, where you're really going to look at it this semester, is in the hydrolysis of ATP and GTP. So you've we've talked about ATP in general biology, hydrolysis for energy. Again, we're not going to talk about ATP as an energy molecule, but rather as one that phosphorylates a different protein. Again, you need water to break this bond so that it can be added to another protein. Um, just making sure I got you everything. Yes, so we will focus on phosphorylation. GTP, the phosphate is not used for GTP to be, let me say that again. Let me write it this way. GTP hydrolysis is not used for phosphorylation, right? So even though you see that GTP is going to GDP and we're losing a phosphate, this phosphate is not put on anywhere. This phosphate will be recycled to eventually make more GTP or GDP. So we're going to talk a lot about GTP aces. Anytime you see ASE, think enzyme. When you think enzyme, think protein. So this GTP cycle is a way to activate and inactivate. Oops, what happened? I touched something. Go back. Activate and, oh, sorry. And in this case, it's the GTPase that's being activated or inactivated. So when an enzyme called GTPase binds to GTP, it's on, it's active. When GTP is hydrolyzed to GDP, so it's lost that phosphate, now this enzyme is off or inactive. So two important proteins you probably haven't heard of before, and they are GAP, which stands for GTPase activating protein. And so just like its name, the protein GAP, it's a protein, helps GTPase hydrolyze GTP. So GTPase without a gap will stay bound to GTP and it will never get turned off. right? So it needs that gap protein to help it hydrolyze to get to that inactive state. Once GTPase is bound to GDP and off, in order to reactivate, you have to exchange, well, we'll say by nucleotide exchange. So there's a protein called GEF. Guanine nucleotide exchange factor. Whenever you see the term factor, it normally refers to a protein. So for GTPase to get reactivated, GEF has to come in and it literally removes the GTP. I'm sorry, removes the GDP and exchanges it for GTP. So this arrow is trying to show you GTP in, GDP out, and now we're back to GTPase binding to GTP, and it's back to the on state. So make sure you have this cycle down. We're going to see it a lot with cell signaling. Okay, one more concept for you that's probably new is this concept of cyclic AMP and cyclic GMP. And these are super important. What I keep doing in must be that part of the screen it doesn't like. Cell signaling. These molecules are something called secondary messengers. You don't have to worry about that term just yet. But what I do want you to understand is how cyclic AMP is made and broken down. Cyclic GMP follows the same pattern. So you take a molecule of ATP, and this is ribo ATP, and there's an enzyme called enediol cyclase. And look at this, you lose two phosphates. So instead of breaking the bond here, where we do for energy, you actually break, excuse me, break the bond here and lose those two phosphates. And then there's this interesting reaction where it cyclizes. So that CAMP, that C stands for cyclic. And that's shown down here. So it cyclizes these bonds. And so this has, cyclic AMP has a role as a secondary messenger. Cyclic GMP is made the exact same way. You take GTP. The enzyme this time is called guanylyl cyclase. And it makes cyclic GMP. This image shows you a little overview. ATP to cyclic AMP. So adenylate cyclase and adenylyl are the same thing. So I usually say adenylyl cyclase. That's just what I'm used to. Means the same thing. And same with guanylate cyclase or guanylyl cyclase. PDE3 is an enzyme called phosphodiesterase. Again, an enzyme. And what it does is it basically um, breaks this cyclic bond, and AMP is reformed or GMP. So this stands for mono, so it only has one phosphate. And if you look at the structure up here, you can see this is cyclic AMP mono, just one phosphate. So when phosphodiesterase comes, now cyclic AMP is gone, broken down, and in cell signaling, that cell pathway would be turned off. Okay, so here are the basic things to know. Central dogma, dehydration synthesis, hydrolysis, the GTP cycle, cyclic AMP and cyclic GMP, and then a little bit about your macromolecules and the concept of genome versus proteome. Nope. And how do I do this? Oh goodness, that's everything. Thanks.